DNA, which stands for deoxyribonucleic acid, is the makeup of the genetic material of living organisms. It is a polymer made up of many nucleotides, which include a phosphate group, a deoxyribose sugar, and a nitrogenous base.
One end of the strand has a 5' end with the 3' end on the other side of the strand. The opposite strand is anti-parallel to the first strand, meaning the 5' end and 3' end are opposite.
The bases are formed from complementary base pairing. Guanine, which is purple, is paired with cytosine, which is yellow, while adenine is pink is paired with thymine, which is orange.
But how does the sequence of a DNA molecule affect all living organism features? There are 3 processes: replication, transcription, and translation. For us to grow, our cells need to divide, which means we need more DNA. We can do this through replication. DNA replicates by semi-conservative replication, which means that one strand of the parent double helix is used as a template to form new strands.
The double helix strand unravels by an enzyme called helicase, which starts from one end of the strand and breaking the hydrogen bonds between the complementary base pairs, making 2 single strands. The single strands will act as templates for the synthesis of new complementary strands. The original strands that will be used as templates are called parent strands.
The free nucleotides roaming around the nucleoplasm bond pairs with the unbonded bases on the parent strands. The DNA polymerase enzyme helps catalyze the bonding between the base pairs to form hydrogen bonds between them. DNA polymerase cleaves the 2 excess phosphates and uses the energy released to link the nucleotide to the new strand.
Transcription is the first gene expression where the encoded RNA is used to synthesize a protein. Transcription occurs in the nucleus, where the DNA is used as a template to form messenger RNA.
Binding causes the DNA to unwind and open. The complementary strands of DNA are now single-stranded. The RNA, which stands for ribonucleic acid, is a single-stranded molecule, meaning only one strand of DNA will be used as a template to form mRNA.
The RNA polymerase is used as a catalyst for this process and moves along the DNA strand. Free RNA nucleotides float into place by complementary base pairing. The RNA links the nucleotides from the 5' end of the molecule to the 3' end. This strand is called the antisense strand, while the unused strand is called the sense strand.
Once it reaches the terminator portion of the gene, the DNA strand and the messenger RNA transcript dissociate with each other. This allows the messenger RNA to leave through the nuclear pore to begin translation.
Translation is the process of protein synthesis in which the genetic information encoded in mRNA is translated into a sequence of amino acids on a polypeptide chain.
The mRNA, which contains codons for the peptide sequence, finds a ribosome and aligns itself. A tRNA molecule appears and forms anticodons based on the complementary base pairing and aligns with its appropriate codons.
Each tRNA carries a specific amino acid according to its bases. Amino acids attached to each tRNA is catalyzed by an enzyme to form a condensation reaction, which joins the amino acids by a covalent bond known as a peptide bond.
The first tRNA breaks its bond with its amino acid to make room for more tRNA molecules to attach and form a peptide bond with the amino acids. The ribosome moves along the mRNA molecule, synthesizing a polypeptide chain until it reaches a stop codon.
Once it's finished, the polypeptide breaks away from the tRNA and is free to roam around the cytoplasm.
We hope you were able to learn about the 3 protein processes that occur in a cell.
Goodbye!